So our next lesson is going to be on synaptic transmission. And essentially all that means is how does the signal from this neuron get passed onto this neuron? Okay, and we had discussed in the previous lesson about how there was two ways that that could happen. It could either happen chemically, which is what you see happening here, because the chemicals will go from one neuron to the next. It can also happen electrically, which would mean there's an actual connection here, a gap junction. But in this case, we're going to be talking specifically about chemical conduction. Okay, so as we mentioned before, this communication happens at the synapse. And the synapse is this area. It's the point of communication between the two neurons or between a neuron and a muscle or gland. So in this case, we can't really see if this looks like a muscle that it's sending the message to. So this neuron is connected to the muscle and it's sending this chemical signal to the muscle to tell it what to do. So whether it's to walk or to remove their hand from a hot object, whatever the message is. And if you look here, okay, so this is the axon that the signal is coming down. Action potential that we discussed in the last lesson is coming down in here into the axon terminal, and then you can see that there's a space. And there's a space between the axon terminal and the dendrite of the next axon, or of the next neuron, sorry. Okay, so there's that little gap there, and that's the synapse. It's essentially the point of communication between those two things. So as I mentioned in the last video, synaptic transmission occurs due to neurotransmitters. And those neurotransmitters are the small chemicals that come from the axon terminal of one into the postsynaptic cell. So we call this the presynaptic cell, and this is the postsynaptic cell. So the neurotransmitter is what actually initiates the action potential on the next neuron. Okay, so the neurotransmitters will leave this presynaptic cell and they will initiate that same action potential that we discussed in the last video into the next one. Okay, so there's a small gap between the axon terminal of the presynaptic cell and the postsynaptic cell, and that area here is called the synaptic cleft. Okay, so the synaptic cleft is that actual gap that's in between the presynaptic cell and the postsynaptic cell, and that's where the neurotransmitters end up going. Okay, so here are the actual steps that it takes to transmit the signal to the next neuron. So when an action potential arrives at an axon terminal, it activates a voltage-gated calcium channel. Okay, so these are right here in green. So these voltage-gated, meaning it has to reach a certain voltage for them to open. And this is the calcium here, the Ca2+, because it's a calcium ion. There's a lot more calcium outside the neuron than inside, so calcium is going to rush in. So just due to regular homeostasis, the fact that there's a lot of calcium outside of the cell and there's not a lot inside the cell, as soon as you open up those gates, calcium is going to rush in. Okay, so calcium goes in. So what happens here is that when the calcium comes in, that the calcium allows these synaptic vesicles, which are right here, these little blue with the dots are all the different neurotransmitters. So the calcium allows these synaptic vessels to actually fuse with the axon terminal membrane. So you can see them moving down, and these will move down towards the membrane, and when they move down towards the membrane, they actually fuse with the membrane here. And when they fuse with this membrane right here, they allow it to open up. And so when that membrane opens up, it allows the neurotransmitters to be released into the synaptic cleft. And you can see that with all the blue dots here. Okay, so all the blue dots are the neurotransmitters, and those neurotransmitters then can be released into this synaptic cleft. Once they are in the synaptic cleft, they will diffuse across it, and they will bind to these little receptors. Okay, so the little receptors are the pink shapes here, and so those are receptors, and they're very specific receptors. They only will receive specific neurotransmitters. Okay. So the neurotransmitters will diffuse across this membrane and they will bind to the receptors that are on this postsynaptic cleft. And then the binding allows these ion channels to open. Okay, so that would be these channels right here. So I'm going to zoom in a little bit here so you can see it a bit better. Okay, so you should be able to see there's little blue dots on the top of each of these ion channels. And that's the receptor, and that's where these neurotransmitters bind to. And when it binds to it, it's almost like a key. So as soon as it binds to it, these channels here will open. And when those channels open, these neurotransmitters are allowed to go from the cleft, and they will enter right into the postsynaptic cell. So when those neurotransmitters come in, that is going to activate this depolarization here, and that will allow the action potential to continue on to the next neuron. Okay, so once you allow these neurotransmitters come in, it causes the depolarization that we saw in the previous lesson, and then the action potential will fire. Okay, so that's what these steps here suggest. So the action potential reaches the axon terminal. So action potential here is arriving. It's arriving at the terminal. It depolarizes this membrane. Once this membrane here gets depolarized, what it does is these voltage-gated calcium ions channels will open. And when they open, the rush of calcium goes into the cell. Once the calcium goes in, that calcium influx, okay, that just means a lot's coming in, the calcium influx triggers these synaptic vesicles, which are these, to release the neurotransmitter. So they travel down towards the membrane, they release the neurotransmitter into the space, and then the neurotransmitter will then bind to these channels here. And in this case, it's allowing these positive ions to flow into the next cell. And once those ions flow into the next neuron, then it's going to allow that action potential to continue. So there's a couple different ways that this can work. So when you're binding, the binding can either stimulate another action potential. Okay, so just like we were showing in the last one, if it actually goes through and stimulates the next action potential, it's called an excitatory effect, meaning it's going to pass on that action potential to the next one, and it's going to continue on down the line. Okay, the other thing it can do is it can actually make another action potential less likely. And if it's less likely to happen, we call that an inhibitory effect, meaning it's going to inhibit the action potential from continuing. So those neurotransmitters binding can do a couple different things. They can either pass on the next action potential so it can continue in the next neuron and continue passing the signal, or it can make the action potential less likely to happen. So for certain chemicals or certain drugs, they want to access that inhibitory effect so that it doesn't allow the next action potential to continue. So when we're looking at the excitatory effect essentially does what we showed in the last diagram. So it binds the neurotransmitters, and that neurotransmitter then depolarizes the membrane of the next neuron. And just like we showed in the action potential from the last lesson, the sodium channels will then open. Okay, so if we have our diagram that's starting to look like this, then sodium channels are opening and the action potential is reinstated. Okay, and remember there's always that threshold that it has to hit. Okay, so as long as it hits that threshold, it's going to continue on down the line and eventually end up hyperpolarizing and end up back at neutral. Okay, so it will pass that on, and it'll continue passing it on over and over and over to the next until it reaches the muscle or the gland that it's trying to affect. Now, what happens with the inhibitory effect is that the membrane potential of the postsynaptic neuron is below the threshold for firing an action potential. So meaning that the neurotransmitter will bind to the target, it'll bind to the next neuron, but it will not allow the neuron to get above that threshold level. So remember, this is our threshold here. And so essentially what is going to happen is that the next neuron, okay, it will be at rest, and it won't make it all the way up to that threshold, and so it won't continue on, and therefore the signal doesn't get passed anymore. Okay, so again, different parts in our body, they want to inhibit different neurons. It's not always a bad thing. Different chemicals in our brain and things like that work that way, but this is two different ways that an action potential can either continue or not continue. And on your worksheet, on your lesson, I put a link to an animation there. It shows how the neurotransmitters jump through that synaptic cleft and bind to the receptors on the postsynaptic neuron. So it just goes through that and it kind of talks you through it. So I would suggest clicking on that and going through that so that you can visualize what we're talking about. So an example of a neurotransmitter is acetylcholine, or you'll see it as ACH. So acetylcholine is a common neurotransmitter, and it's needed to basically do almost everything in our body. So for us, it triggers muscle contraction, and therefore if it triggers muscle contraction, it's the thing that helps us move, it's the thing that helps us talk, it's the thing that helps us all of our behavior. It also stimulates a lot of hormone secretion, so different types of hormones that we have in our body will help to trigger that. It's involved in a lot of things in our memory as well. So memory, learning, anger, aggression, attentiveness, all those types of things are all triggered by this neurotransmitter acetylcholine. And so scientists have realized that acetylcholine releasing neurons, so those are the neurons that actually release the acetylcholine, they happen to degenerate in people with Alzheimer's. So that's how they believe that Alzheimer's occurs, is that when their memory and their learning starts to decrease, that's because these neurons that actually release acetylcholine are starting to degenerate, and that just means they start to shrink and they're not able to do their job anymore. So acetylcholine can have both excitatory and inhibitory functions. That means they can either speed up or slow down nerve signals. So if they want it to continue or if they want it to block something, they can do both of those things. But the majority of what it does is excitatory. So learning, it wants to be able to pass those messages on. Memory, muscle contractions. Okay, we want the message to get all the way from our brain to our spinal cord and to the muscle so that it can actually contract. So those would all be excitatory. They've also used the acetylcholine, its ability in different types of poisons throughout history. So curare was used as an arrow poison by some indigenous peoples of South America. And what it does is it blocks muscle contractions and produces paralysis by competing directly with acetylcholine for binding sites in the muscle cell synapses. So now that they know how acetylcholine works and how it can bind to the receptors and then allow that message to continue, what they found now is that this poison here, they have made it so that it actually competes with the spots that acetylcholine normally wants to connect with. And so those binding sites then are occupied by this poison, and so it causes paralysis in people because they can't have their muscles contract anymore. Nicotine also binds to acetylcholine receptors. So instead of the acetylcholine being able to bind it, nicotine does instead. But nicotine actually acts as a stimulant by turning the receptors on rather than off. So it allows you to continue with that stimulus as well. So these are the steps involved in acetylcholine and how it binds to the postsynaptic neuron. So first, acetylcholine will bind to its receptors and the sodium ion channels open. Okay, so the acetylcholine will bind to the receptor, which is right here, this purple thing. It will bind to the receptor, and in that case, then the sodium channels open for that second membrane. And those sodium ions will then rush into the postsynaptic neuron, which is this one, which causes depolarization. So again, that charge reversal that we talked about last lesson. The reversal of charges causes an action potential to move down the postsynaptic neuron. Okay, so the charge is then going to be reversed this way, and the action potential will move down the postsynaptic neuron and continue on that way. And again, that's if the threshold is reached, so we have to reach that minimum amount. However, in order for the postsynaptic neuron to repolarize, okay, meaning, remember, it has to depolarize, which will pass on the signal, but then it has to repolarize to get back to resting membrane potential. So in order for that postsynaptic neuron to repolarize and receive other nervous signals, because if it doesn't repolarize, it can't receive the next signal. What it does is it releases an enzyme called cholinesterase, and it's also called acetylcholinesterase, and that's an enzyme that actually breaks down the acetylcholine. So down here at the bottom, okay, so you can see your acetylcholine here, that's your A and your CH. So the acetylcholine here will then get broken down by this enzyme here, which is acetylcholinesterase, and it will get broken down into acetate and choline, and then those will eventually, the choline will make its way back into the cell, connect with acetyl-CoA. Okay, so remember that molecule way back when from cellular respiration. So from the mitochondria, the acetylcholine, or sorry, just the choline will combine with the acetyl-CoA to make acetylcholine. And once it goes into the synapse here, then this enzyme will break it down so that we can reuse it. Okay, so once the acetylcholine has been destroyed, okay, so once the acetylcholine is destroyed, the sodium channels will then close on this membrane and the neuron becomes repolarized. So once we destroy that enzyme again, once we destroy the acetylcholine, then the sodium channels are able to close and this area is repolarized. So it's ready to receive the next signal transmission. And again, these things all need to happen quite quickly because we need to receive those signals, within milliseconds so that we can react accordingly to how our brain wants us to react and also how our muscle and glands need to react. So some other common neurotransmitters, there's tons and tons and tons of them, but we're just going to focus on a few. So one is serotonin. Serotonin, I'm sure you've heard of before. It's involved in many different brain functions. It's released by a small number, and again, a small number in terms of our brain is a few thousand neurons in the brainstem. And these neurons are connected to at least 40 million other neurons, so it's a very, very important neurotransmitter in our brain. Any imbalances in serotonin, as well as norepinephrine and dopamine, if you've heard of those, happen to be linked to depression. So anytime that they're talking about a chemical imbalance when it deals with different types of mental health or depression, a lot of times it has to do with these actual neurotransmitters not being able to bind or something blocking them. So if you have any imbalances in your level of serotonin, oftentimes it can be linked to depression. So what antidepressant drugs do is they act on these pre- and post-synaptic neurons to alter the rate of the neurotransmitter breakdown. So how quickly are those neurotransmitters being broken down? Is there an imbalance in that? And a lot of those drugs will act on those areas. The next one that we're going to look at is dopamine. And dopamine is critical for executing smooth, controlled movements. So those would be, any movement that you would do during exercise or things like that. It's critical for executing smooth, controlled movements. Lack of dopamine is a cause of Parkinson's disease. So oftentimes we can do things very slowly and controlled, whereas people who have Parkinson's are not able to do that. So they tend to shake a lot. They're very jittery muscles, and that's because their muscles do not have they don't have enough dopamine to make those controlled movements happen. A disruption of flow of dopamine may cause schizophrenia as well. They believe that a disruption to the dopamine flow can actually lead to schizophrenia. Dopamine also produces a feeling of bliss or painlessness. So during exercise, when they talk about that runner's high, that's often what they're talking about, is a release of dopamine into your body when you're exercising, and that gives you that feeling of euphoria. So many drugs will mimic or stimulate the release of dopamine as well. So they'll capitalize on that. So they'll either mimic how dopamine makes you feel, or they're actually stimulating the release of dopamine to give you that feeling of bliss. Overstimulation of dopamine receptors on postsynaptic neurons can cause the neuron to decrease the number of dopamine receptors. So in other words, you get to become drug tolerant. Okay, so you have to have, when they talk about drug tolerance, they mean, the first few times you have it will affect you a certain way, but the more you use it, the more that your tolerance increases, and you need the same amount you need more and more and more of that drug to have the same effect. So that's what's happening here, is that if you overstimulate those dopamine receptors, meaning you're releasing the dopamine due to the drugs that you're taking, it can cause the neuron to actually decrease the number of dopamine receptors. So those receptors start to go away, meaning you need to have more and more of the drug to get the same results that you did the first couple times you tried it. Your body will also decrease the amount of dopamine it makes, so responsible for withdrawal once drug use is discontinued. So once you stop taking the drug, your body will also decrease the amount of dopamine it actually makes. So you were pumping it out a lot when you were using the drugs, and your body will then decrease it, and that happens when you start to go through withdrawal once your body stops using the drugs. Another one that we will look at briefly is epinephrine, and you may have heard of epinephrine before. It has an excitatory effect in the parasympathetic nervous system, but it can be excitatory or inhibitory in the central nervous system. So if you're not sure what those two are, please go back to your first note on this lesson. Epinephrine is involved in cardiac function. So if it's had the excitatory effect, you have an increased heart rate, and it can influence metabolic rates as well. So the epinephrine also can have a very excitatory effect. So again, drugs are capitalizing on that, and they're making drugs that will react like epinephrine does to give you kind of that excitatory effect. We have neurotransmitters called endorphins, and endorphins is what I was discussing earlier. When they get released, they happen during pleasurable exercise. So eating something that is, one of your favorites, or sexual intercourse will also lead to a release of endorphins. Any type of physical stress, so after extended physical exercise, then they can have an opiate-like property. An opiate is like a painkiller type property, so it reduces pain and it induces euphoria. So that example of that runner's high. So it gives you that feeling of overwhelming kind of bliss, and you're feeling good right afterwards. And that's it for synaptic transmission. So overall, you should have an idea of how neurotransmitters gets passed from the presynaptic cell to the postsynaptic cell, how calcium influences how those vesicles are released, and different types of neurotransmitters that we discussed here, and just kind of a general overview about what they are and how they work and things like that. So again, on OneNote, I've put up some videos so that you can have a look at some visuals. Again, please look at those so that you have an idea of how this all works, and if you have any questions, please bring it to me on Monday. Thanks.